Speaker is an author. He wrote a book called Evidence of Harm. If you want to get upset and have your blood boil over, this is the book to read, let me tell you. You cry, you don't even laugh too often. It is an amazing work of love and dedication. And when you sit back, I sat there, Charlie Brown always sits there and looks at me and says, how can I get Andy Landerman and Mike Margolis, who are in two different political worlds, one a staunch Democrat, one a staunch Republican, to agree on anything. And I sat there and I said, I'd like to take this book and place it someplace on a number of politicians. And I wouldn't want to ruin the book, though. The other aspect was Charlie looked at me and says, it's in both parties, Mike. You can't even take the blame for this one. The Evidence of Harm opens your eyes to the reality of politics, medicine, and pharmacology today. It's my pleasure to introduce Mr. David Kirby.
It's really great to be here. I like speaking to different groups. Normally when I speak, it's very often to autism groups, and I'm speaking to parents. And the more I can speak to people who aren't necessarily directly affected by this subject, the better. And the two best professions that I've been speaking to so far are dentists like yourself and also chiropractors for slightly different reasons. I think chiropractors in general tend to be very, very anti-vaccine, and my guess is that your main concern here today is the mercury that's used in amalgams. And mercury in amalgams most definitely works into this story, and I'll be talking about it quite a bit. But mostly I'm talking about mercury in vaccines as a possible cause of autism. That's what my book was based on. But really, and I think as we move into the next couple of years, this whole idea of a variety of environmental triggers that may be causing a variety of outcomes in a large number of American children with certain genetic predispositions. And one day we may actually be talking about autisms. And we may one day find out that things like ADD and ADHD are actually all part of a spectrum of environmental toxicants that somehow made their way into certain children at a certain point in their development and allowed things to happen such as inflammation of the brain, such as disruption of the nervous system, such as immune problems. And you'll see that mercury can cause a lot of these things. I will be talking mostly about science today. There is a whole nother part of the book that's all about politics, and there's a whole nother part of the book that's all about the law. Very, very fascinating. That is going to be dealt with, I'm happy to announce, in a movie that's being made by Participant Productions, who just did Al Gore's film, and they did Fast Food Nation. Last year they did Syriana and North Country. Thank you. The company is called Participant Productions, and they're really terrific, and they just had a meeting of their board of directors and voted to proceed with the movie. The script is now in its second draft, and we hope to be casting for directors in about a month or so, which should make a huge amount of news. Now, I know that the company has been brought under tremendous pressure from very, very powerful people to not make this movie, and they went ahead and made the decision that they're going to continue. So if you ever get a chance, send off an email to Participant Productions and tell them that they're doing the right thing. This story needs to be told, and we need to find out what's happening to our children. And I'm not here to say that mercury causes autism. I'm not here to say that thimerosal, this mercury preservative in vaccines, causes autism. But it is clear that our kids are in trouble. 1 in 6 kids in this country has some form of learning disability. We also happen to know that 1 in 6 kids in this country is born with mercury levels already high enough to cause neurological damage sometime in their life. That's before they take their first breath. So those are prenatal exposures. American women are exposed to huge amounts of mercury. Most of that ends up in the fetus. And we're talking mostly about organic mercury, and very, very recent studies show that that can lodge in brain stem cells, developing stem cells of the brain, and essentially shut them down. So as we have this debate, and you probably know we are having this debate in the country, there are a lot of people who do not want this theory to pan out. There are a lot of people who think that autism is not even epidemic, that the new case numbers are merely a product of better reporting. And what we have is somewhat of an impasse in this country. We have most of the medical establishment saying, this has been disproven, let's move on. But then you have a handful of researchers who are very, very dedicated, and literally tens of thousands of parents who are saying, not so fast. If mercury doesn't cause autism, why is my kid so sick? Why does my kid have so much mercury bound up into his system, usually his, sometimes her? And why, when we try to treat mercury toxicity in our children, do at least some of them seem to get better? And where we are now is an argument between people who look at large population studies and say, we've looked, there's no association between mercury and vaccines and autism. And then people who do the actual bench science, people who look at the kids, who do animal models, who do what we call biological studies, and they say, no, there is ample evidence to support this link. So that's sort of what we'll be looking at today. I mentioned that we may one day be talking about autisms, and right now when we use the word autism, we use it very, very loosely. And unfortunately, the English language is very deficient in trying to describe these kids and what they have and what's causing it. But we use a term called autism spectrum disorder, and roughly speaking, there are three main categories. There are some others that fit on this spectrum. There's what we call full-blown autism, classic DSM-4, and that's the one where they really, the kids are in very, very bad shape, can't talk, have problems even walking, the repetitive behaviors, the self-injurious behaviors, the spinning. Somewhere in the middle is what we call pervasive developmental disorder, not otherwise specified. And then the highest functioning form of ASD is Asperger's syndrome. Now, some people speculate that perhaps people with Asperger's don't have the same disorder as these kids with autism. Asperger's, as you may know, is generally typified by extremely intelligent, high IQ, and very, very systematic and orderly ways of thinking. People with Asperger's are usually incredibly good with numbers and computers. Then when you look at the kids, for example, in my book, who can't even really go to the bathroom properly or tie their shoes, it's hard to understand how a child that bad off could somehow grow up into an adult that can go to school and get a graduate degree and work at Microsoft and go on TV or write a blog about their experiences. My guess is that those two people have two different disorders, even though there are some crossovers. And it's possible, and this is one of the things that we just need to really look into, that some people have a purely genetic-based form of autism, that they were going to be autistic, what we call autistic, no matter what. But a majority, somewhere between 60% or more, of kids who have autism regress. They were born, they were perfectly normal, they met all of their normal milestones, and then somewhere around 1 or 2 years of age, they began to regress, some of them really, really quickly. And that doesn't seem to be the case with the older people with autism. So we may be talking about autisms, we may one day call them autism type 1, type 2, type 3, or we may come up with a whole new name for what we today are calling autism, something along the lines of some type of acquired neuroimmune disorder. Because what we're seeing in most of these kids is they have terrible immune problems, they have terrible neurological problems, and they have inflammation. And it all seems to be related to a autoimmune response to some type of toxicant that they've been exposed to that causes these immune imbalances, that causes secretion of cytokines, causes the Th1, Th2 balance to become disrupted, and causes neuroinflammation. Their brains grow really, really fast. These are the main forms of mercury that we are exposed to in our modern world. And when I say our modern world, all of them are on the increase. And we'll see later about the environmental exposures that we have in this country. But basically speaking, there's elemental mercury. We used to break in the thermometer and roll around in our hands. As you all know, it's in dental amalgams, and it's in lights and batteries and everything. And whenever these break or get drilled out of your mouth, we inhale mercury vapors from them. There is inorganic mercury, which comes out of the most common sources, coal-fired power plants, and we'll take a look at coal and what it's doing around the world. Everybody thinks coal is a local issue. It is not, folks. Coal is a global issue. And what's happening in China affects kids right here in Arizona, believe it or not. It also comes from volcanoes and wildfires, and we'll take a look at wildfires towards the end when we look at environmental exposures. But what goes up must come down. So whenever there's mercury in the air, it comes down and it settles on the ground, on forests, on trees, on leaves, grass, etc. It will stay there forever until there's a fire. And when a forest fire goes up in flames, it emits sometimes 20, 30, 40 years of mercury accumulations in 1 minute. And there is more mercury in an acre of fuel than there is in probably, well, I don't know how many dental amalgams there are in this room, but in a typical room, we're talking about ounces of mercury for every acre of forest that goes up. And again, if you're a pregnant woman living downwind from one of these fires that tend to burn for days, you're exposing your fetus to massive amounts of mercury. There's two types of organic mercury. There's methyl mercury, which is found in fish, and we eat that. And that is converted from inorganic mercury that comes out of these coal-fired power plants. It goes into the waterways, converted by microorganisms, and then moves up the food chain until it lands on our dinner table. The sushi fish actually are the highest ones: the tuna, the mackerel, the shark, the swordfish. Very, very high in mercury and getting higher all the time. Then there is ethyl mercury, which also is organic. Now, organic is lipophilic. It binds with fat, which makes it extremely dangerous. The body, because it binds with fat or proteins, the body generally recognizes organic mercury as a nutrient. So if you're not pregnant, it will try to send that nutrient up across your blood-brain barrier into your brain. And if you're pregnant, unfortunately, it will send it right past the placenta, and into the fetus, where it accumulates. And that's why so many children are born with such high levels. The main form of ethyl mercury is thimerosal. This was invented by Eli Lilly in the 1920s. Began to be put into vaccines as a preservative in the 1930s so that they could sell vaccines in 10-dose vials. Every time you stick a needle through that rubber seal, you might introduce harmful bacteria or fungi. So they needed something in the solution to prevent contamination. Mercury was cheap and abundant, and Eli Lilly had the patent. So they started putting into vaccines. Oddly enough, the first case of autism was not identified until 1943, about 4 or 5 years after we started using mercury in vaccines. No idea if that's a connection or not, but it is a pretty interesting coincidence. Also, and we start looking at environmental exposures to mercury, because so much ethyl mercury is used in the manufacturing process by pharmaceutical companies, it too is now a pollutant in our waterways. We are finding fish with ethyl mercury pollution in them, and particularly in states like New Jersey, where we'll see not only is there a lot of mercury, but it has the highest autism rates in the country. It is packed full of pharmaceutical industries, and they have often been cited for spillage of ethyl mercury from their plants. Now, these are the numbers, and this is such a hot burning debate even still, and I have to admit, my book has been out for 2 years, and I'm a little shocked and dismayed that I still have to argue that there actually is an autism epidemic, that the numbers actually are going up. And the people who argue against this are usually the people who are have a vested interest in defending vaccines and the use of mercury in vaccines. And the only way to really put this argument to rest is to put forth the argument that there is no autism epidemic, that the numbers are not going up, that the numbers have always been what they are. We're just better now at noticing it. The reason that argument is so important, because that would mean that autism is purely a genetic disorder. There's nothing we can do about it. It's part of the human condition. It's always been with us. It will always be with us at that rate of, as you can see, up until recently, the estimated rate was 1 in 166, even though in the 80s and 90s it was much lower than that, and we saw the rate continue to go up and go up and go up. Well, you cannot have a genetic epidemic. Therefore, if the numbers are truly going up, and it's not just better reporting, there must be something else happening to our kids. Yes, they have some type of genetic predisposition, but a good percentage of them encountered some type of environmental trigger at a very opportune time, either prenatally or, say, in the first year of life or two, that set them off. And we are still having this debate because once you prove that the numbers actually are going up, then you, by definition, prove that there is an environmental component to autism. And once you start looking at environmental components, you have to look at mercury. And once you start looking at mercury, what do you have to look at? Vaccines, dental amalgams, fish, all of the ways that we're exposed to them. And there's been a huge reluctance to do that. So a lot of epidemiologists are still saying, no, 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 we simply broadened the criteria, we added on PDD, NOS, and Asperger's syndrome onto what we call ASD, and that's the reason the numbers went up. Well, I have a lot of arguments to that, and we'll be talking about them. But if we look, for example, at the US and the UK just stopped using mercury in their vaccines about 2 years ago. Their ASD rate was about the same as ours, about 6 per 1,000 kids. However, of those 6, 4 were somewhere on the spectrum, and only 2 had the full-blown hardest, most severe form of autism. Whereas in the United States, we have 6 per 1,000, or did. And of those 6, 4 were full-blown DSM. So at least from the statistics that we had at this time, it could be argued that the rate of full-blown autism, thank you, in the United States is twice as high as the rate in the UK. And guess what? The amount of mercury we give to our kids, or were giving to our kids, was roughly twice the amount that they were giving in the UK. So it's possible, if mercury is implicated in this spectrum disorder, that it really, the dose does make the poison. So that because we gave American kids, in other words, there's a certain threshold to get to this ASD spectrum, but we gave so much more mercury to our kids that we ended up with 4 out of 1,000 with full-blown autism. In the UK, they ended up with 2. And then there's countries like Denmark where the rate is about 8 per 10,000, or 1 in 1,300, much, much lower than in this country. And you have to ask yourself, well, if autism was purely genetic, and if the case numbers going up is just a function of better diagnostics and better healthcare, Denmark, a small, efficient, socialized country with universal access to healthcare, how is it possible that we are 6 times better at diagnosing ASD in this country than they are in Denmark? It's a laughable question. I don't think we are. I think we have more cases than they have in Denmark. What's different? We use thimerosal in our vaccines. Denmark took them out of their vaccines in 1992. And it doesn't stand to reason that the United States genetic makeup is so radically different than that of Denmark, particularly when we see autism affecting all races and all ethnicities across the board in this country. There's got to be some other explanation. So then we want to look at what happened to our kids, what's different about our kids, what happened in the United States. And prior to 1988, there was only one vaccine in this country that we gave to kids with mercury in it. That was the DTP. We gave 3 shots for a total of 75 micrograms. That was spread out over a fairly long period of time. I haven't seen the actual schedule, but I think the first shot you got around 6 months to a year, and then you get another one maybe at 18 months, and maybe another one at 2 years. In 1988, we added a new vaccine onto the schedule, HIB, Haemophilus influenza B, that called for 4 shots with 25 micrograms each. And also, right around that time, we added a 4th DTP shot. It's now called DTAP. So by 1989, roughly, kids were now getting 200 micrograms of mercury in their vaccines, starting at 2 months of age. Whereas prior, they were only getting 75 micrograms of vaccine, starting around 6 months to a year. Then in 1992, we added another vaccine onto the schedule, hepatitis B. There's a lot of uproar right now about the HPV vaccine, vaccinating 13-year-old girls for human papillomavirus. People say it gives teenage girls a license to have sex, but nobody bothers to point out that we're vaccinating newborn babies against hepatitis B, which is a sexually transmitted disease or a disease of IV drug users. And I don't think there's a lot of IV drug use going on in nursery school. But it's interesting how nobody says a word about this, but vaccinating a 13-year-old girl for HPV is so controversial. Without taking, nobody wants to get hepatitis B, and we should all be protected against it. I personally question the wisdom of a newborn needing a hepatitis B shot. And that's 3 shots with 12.5 micrograms each. So by 1992, 93, 94 really, it takes a while for compliance rates to go up, kids were now up to almost 240 micrograms of mercury on top of all of those prenatal exposures that their mother had. And that would include mercury in amalgams, mercury in fish, mercury in a substance called Rho-D immunoglobulin that Rh negative women get. mercury in the flu shot that pregnant women get. So this was just the postnatal exposures. We went from 75 micrograms to 240 micrograms in very short order. Nobody ever stopped to add up the total mercury burden. At the same time, when we look at least the United States Department of Education data, there was a huge spike, and the greatest increases were kids born between 1987 and 1992. That's exactly when we added all the mercury. So this is what's called an ecological association. It's not proof there are other factors at work, but it sure is an interesting coincidence that just in the same years that we added these new vaccines with all this mercury in it, those very birth cohorts later on went on to develop far more autism than anybody else had prior. Now, one reason this gets so complicated is right in this time, right around 1992, states started adding PDD and Asperger's syndrome into their count of autism, their ASD count. And sure enough, the rates started going way up. So when people say, oh, it's just better diagnostics, it's right criteria, that actually does explain some of the rise, but it doesn't explain all of the rise. It doesn't necessarily have to be all one or all the other. The world is more complicated than that. I believe both factors are at play. And when we look at California, which only tracks full-blown DSM-4, it still goes up and up and up, as you'll see. So you can't possibly attribute California to better diagnostics or wider criteria, because they never existed. Now, this study came out last week. You may have heard about it. I understand it was all over the news. I got an email from somebody saying, why aren't you talking about this? You'd have to be on a remote Caribbean island to not know this was happening. Guess where I was last week? It's called Culebra, and it's off of Puerto Rico, and if you ever have a chance, go. Because I heard about Anna Nicole Smith, and that was it. That was the only news of the world that filtered through to us. But this story, of course, was burning up the airwaves. This new study that came out that showed that we now have 1 in 150 kids with autism in this country. And this was an unusual study. I think it took people by surprise. I think a lot of people are on both sides are scratching their heads over, and no one really knows quite what to make of the numbers and what they're telling us. But there are some very interesting things. What the CDC tried to do was to show that there really was no increase between kids born in 1992 and kids born in 1994. The other interesting thing is they made this sound like this was brand new data, that we just found this out and everything. These were studies that went back to 2002 and 2000, looking at 8-year-olds. So this is really, really old data. These are kids who got the full amount of mercury. In fact, if we look at the kids born in the 1992 birth cohort, they probably, on average, received less mercury than kids born in the 1994 birth cohort, because compliance rates take years to catch up. So hepatitis B was still going up, and HIB was still going up. And when we, meaning Safe Minds and the groups that I work with, took the numbers and looked at they had 6 sites that they tested in 2000, and then 6 sites that they tested in 2002. And when you compare those 2 sites, there was an actual increase of 10%. So the rates were actually going up in those sites. But the most interesting thing, I think, out of this study is the range, the differences. They're now looking at 12 sites around the country, and most of them range from somewhere between 5.2 to 7.6 per 1,000 kids. Well, that's actually a pretty big range. That's a range of 46%. So it may not sound like much, but one state obviously has a lot more autism than others. Then we have these 2 outliers, Alabama and New Jersey. And look at this. New Jersey has 3 times the rate of autism as Alabama. And everybody is at a loss to explain that. These are cases where the CDC went back and looked at medical records and interviewed parents and talked to doctors. So this is not just simply reporting. This is actual epidemiological studies where they go and look for these kids. And they found 3 times as many kids in New Jersey as they did in Alabama. Now, are doctors in New Jersey 3 times better at diagnosing than they are in Alabama? I don't think so. And it's not a function of just better services, because these are not based on educational data or special ed. These are actual diagnoses, whether you're homeschooled or in an institution or whatever. And still the rate is so much higher in New Jersey. Well, then it probably isn't a genetic disorder. there's something else going on. And one possible explanation, again, is mercury. And when we look at New Jersey, we find lots and lots and lots of mercury. When we look at California, we find lots and lots and lots of mercury, as we'll see. California also has one of the highest rates in the country. And again, that 10% increase from 1992 to 1994, that's when compliance was going up further. What would be really interesting if they could go back and look at, say, the 1988 birth cohort, or the 1985 birth cohort, or, for that matter, the 2006 birth cohort. Why they chose 2 years in the peak of thimerosal exposure has raised a lot of eyebrows. I would love for the CDC to go back to these same sites and look at kids in the 2000 and, let's say, 4 or 5 birth cohort and see if the case rates have come down at all. Now, if these numbers keep going up and up, like they said, then there should be the same amount. We're now at 1 in 150 children. That's about 1 in 94 men, because the rate is 4 to 1 boys to girls. So there should be 1 in 94 men, my age, everybody's age, in this audience. You should be able to go out, pick 1,000 men at random, and find out that 6 or 7 of them are somewhere on the autism spectrum disorder. They're not there. We've tried to find them. People have gone out to look for them. There are adults with autism, but they simply do not exist in anywhere near the same numbers as children. This is a disease of young people. And by the way, most of these kids have not turned 18 yet. Most of these kids are probably approaching around 12, 13 right now, the bulk of them. Some of them are old enough to start hitting 18, and when they do, that's it. Their parents are tapped out, folks. They're getting on Social Security. The taxpayers are going to foot the bill, and we're talking about millions and millions of dollars for each person for the lifetime of care. So if the American public has not quite woken up to the fact that we have an emergency on our hands, when the tax bills start coming due, you can bet everybody will be saying, where are all these kids coming from? And they weren't there 10 years ago. They weren't there 20 years ago. They weren't demanding the services that they need today. And I mentioned California. This is the California numbers for DDS. They have them by age group, so I broke them down age group by year. So that's the average per year for those age groups. And if you look at the 6 to 9-year-olds, there's 2,100 kids in each age group from 6 to 9 with full-blown autism registered in the California schools. If we look at, say, the 14 to 17-year-olds, there's less than half that number per year. So these are kids, these 14 to 17, they're in high school, they have full-blown autism, they can't speak, they can't read, they can't write, they can't sit in a chair, and yet they're about to graduate at high school and nobody ever said, maybe you have a disorder, maybe you should go get an evaluation. Nobody said anything to their parents. If you believe that this is true, that we're better at diagnosing full-blown autism, and by the way, you cannot miss full-blown autism. You can see it from space. These kids, they're unmistakable. And yet, people would have us believe that there's something like 4,000 high school kids with full-blown autism in California completely undetected, completely undiagnosed. Not getting any services at all. Why would parents of 6 to 9-year-olds be so much more savvy, to get their kid into special education than parents of 14 to 17-year-olds? It doesn't make sense. There are more 6 to 9-year-olds with autism in California than there are 14 to 17-year-olds, period. And that means the numbers have gone up, and that means there's an environmental component to autism. And yet, we still have to argue that there is against the argument, that there is no epidemic. And for medical professionals to try to explain this away as a mere artifact of reporting, I think is a complete abdication of their professional responsibilities to take care of our children. It would be nice to just think that this was not a problem, that it's always been with us and we're just better at noticing it, but that is on its face an absurd statement. And it does a huge disservice to American children. And one day they're going to have to eat their words, because autism is going up. This is the schedule that children received in this country. As you can see, at birth, they got their first shot, 12.5 micrograms. This was up until just a couple of years ago. Depending on your body weight, the EPA says you should not receive more than 0.1 micrograms of mercury per kilogram of body weight. So that newborn getting 12.5 micrograms on average would be 35 times over the EPA limit. If that child weighed only 4 pounds, you double it, and that child is now 70 times over the EPA limit. At 2 months, they came back in for another 3 shots, 62.5 micrograms. That's about 138 times over the limit. Now, to put that into perspective, for me to receive that equivalent amount of mercury, I would have to be injected with 1,300 micrograms of mercury, 1.3 milligrams. That's a visible amount. You could see that on a spoon. And if you think that I would allow myself to be injected that, you're crazy. And yet, we were doing this into newborn babies, and we still are with the flu shot, and pregnant women. And then this went on. At 4 months, they got another 50 micrograms, 6 months, 12 months, et cetera. So as you can see, these kids were constantly being hit with mercury, with very large bolus doses of mercury, well in excess of EPA safety regulations. And the theory is, and we'll look into it, is that a certain percentage of these kids were simply unable to process all that metal. Most of us, when we're exposed to heavy metals, we get them out of our bodies through a complex system of proteins that are sulfur-based. And what we're finding is kids with autism lack these proteins. Glutathione is a very common one. If you are exposed to heavy metals and you don't have the ability to excrete them, it's going to go somewhere. It's going to accumulate. That's why this is so important. Every microgram counts. If you're a poor excreter, every microgram you're exposed to is a microgram of burden, body burden, that your body is going to hold on to. This is the actual bottle of thimerosal. As you can see, it has a Jolly Roger on it. Why you would inject that into a newborn baby, or anyone for that matter, in any amount, seems crazy to me. We know how deadly mercury is. And I mentioned the flu shot, and I was shocked when I sat down and added up the amount of mercury we're giving to American kids today, right now, and for the past couple of years, recommended on the CDC schedule. The flu shot is now recommended for pregnant women, for infants at 6 months, 7 months, and 18 months of age. We've never given a prenatal exposure of mercury in this country, with the exception of Roe-D, which was 15% of pregnant women. We're now giving mercury shots. I mean, I think the compliance rate is something like 50, 60% of pregnant women are getting the flu shot. That's 25 micrograms of mercury going directly towards the fetus. The partition rate is something like 70 or 80% to the fetus, the rest to the mother. It's a cruel joke of nature. You would think it would be the other way around. A doctor once told me, and it's obscene, but it illustrates the point, that the best way for a woman to detoxify from mercury is to have a baby, because the kid will absorb most of the mercury. It gets absorbed in the brain stem cells and in the liver. So we have 25 micrograms exposure prenatally. We have no idea. By the way, if you look at the flu shot, if you look at the insert on the label, it says this product has never been studied for reproductive health. The effect of the fetus is unknown and should only be given to pregnant women when absolutely necessary. But you can walk down to Walmart and they'll gladly give it to you. I have nothing against the flu shot, but we shouldn't be giving it to pregnant women when we haven't fully studied the effects, if you ask me. 25 micrograms prenatally. At 6 months of age, these kids get 12.5 micrograms from the flu shot. At 7 months of age, they get another 12.5 micrograms. At 18 months of age, they get another 25 micrograms. And then there's residual amounts in most of the other vaccines they get, about another 10 micrograms. And remember, if you have an efflux disorder, every microgram counts. So kids in the United States today are getting 85 micrograms of mercury by their 18th month, by 18 months. And you have to remember, in the UK, the maximum exposure was 75 micrograms. So we are now in excess of what the UK was giving at the peak of their thimerosal exposures. And yet, medical professionals will tell you that we don't use thimerosal anymore, that American kids aren't exposed to mercury in vaccines anymore. 85 micrograms is a lot of mercury for anybody. What's interesting is the companies that make thimerosal warn their lab workers against exposure to it. And they say if you're exposed to this problem, some of the symptoms that you may see, and you can see these, and I don't know how much you know about autism, but emotional disturbances, spastic movements, incontinence, groaning, shouting. If you have a kid with autism, most likely he will display a lot of these symptoms. So thimerosal can cause a lot of behavioral problems and symptoms that are somewhat similar to what we see in autism. Also, if thimerosal is so safe as they say, why did the state of California list it as a known reproductive toxic substance? And they say that the evidence that it causes reproductive toxicity is clear and voluminous. So the state of California recognizes, which by the way has banned thimerosal from vaccines for pregnant women and children, this stuff can really harm the fetus, and it can harm children, and yet we continue to inject it into our children. Now, back in 1999, the government issued a statement saying, guess what, we finally added up all the mercury we're giving your kids and compared it to their body weights and looked at the EPA limits, and we're way over the limit, and we think mercury should come out of childhood vaccines as soon as possible. That was July of 1999. We're now going on 8 years, and it's still in there. That's not as soon as possible, as far as I'm concerned. But when that announcement got made, a number of parents around the country, and they're in my book, and they belong to a group called Safe Minds, they started doing research on their own. First thing they did was add up the amount of mercury their own kids got in vaccines, and then they went to the literature. They wanted to see, well, what are the symptoms of mercury toxicity, particularly mercury toxicity in children. And they found this disease called acrodynia, pink disease. It was around in the 30s and 40s and 50s. And it turns out it was caused by inorganic mercury in teething powders that we were giving to children. It wasn't so common in this country as other English-speaking countries. And what was most interesting about acrodynia was that only 1 in 500 children exposed to the mercury came down with the disease. Again, suggesting a strong predisposition, a genetic predilection to have some inability to process this mercury that they were exposed to. What's also interesting is the symptoms of acrodynia. You had the most common was a weepy red rash on your hands and feet, kind of like a very severe eczema. And by the way, I believe that mercury exposure can cause eczema. But the other symptoms were just remarkable: poor eye contact, poor coordination and muscle control, lethargy, repetitive behaviors, rocking, spastic movements, emotional outbursts, et cetera, et cetera. And when you read the accounts of children with acrodynia, you really think you're reading a textbook about autism. And one woman from Australia who now is able to describe what it was like to have acrodynia, and she describes mercury poisoning from the inside out, and it's really fascinating. And again, the rocking, the spastic movements, the not wanting to be with other children, the constant pain, crying, sleeplessness, all the things that we see in autism. Now, people say, well, acrodynia is not autism. Autism is not mercury poisoning. It's very important to remember there are many, many different kinds of mercury poisoning. There was Minamata disease in Japan. There was the Iraqi grain incident. There was Mad Hatter's disease. My friend grew up in Danbury, Connecticut, and he remembers old people walking around town with what they called the Danbury shakes, because it was the hat-making capital of the country. They used a lot of mercury in the felt process, and these old people would have basically Parkinson's, but they were also prone to emotional outbursts and very, very antisocial, and they would not make eye contact. And my friend was always very impressed by that. But that's not autism. So there is no one type of mercury poisoning, and what these parents put forth is that autism is a novel form of mercury poisoning, a new type, but that it depends on the type of mercury, the amount of mercury, the route of exposure, the age of exposure, your body weight, and probably your gender. Because there's evidence, as we'll see, to suggest that testosterone and mercury are very, very synergistic. And that's where I'm going now. I'm going to talk about some of the studies that have come out that do support this hypothesis. Then we'll look at some of the epidemiological studies that have been put out to sort of oppose it. And what you'll notice is that most of the supporting evidence is biological in nature. It's test tube data, animal models, clinical studies, et cetera, whereas most of the evidence to oppose it is epidemiological in large computerized population studies without ever looking at a single kid with autism. And I think to resolve this and to finally answer this question, we are going to have to look at kids with autism. We're starting to look at kids with autism. What is different about this child than this typical child? What's different about their blood, their nervous system, their brains, the way their immune system works? What are the differences, and what could possibly explain that? That's going to answer this question far more efficiently than very, very large population studies. Now, I mentioned testosterone. This was done by probably someone most of you in this room have heard of, Dr. Boyd Haley from the University of Kentucky, a leading toxicologist, and he took vaccine ingredients and wanted to see what happens when you mix them together in different combinations. And what he found out was these are the control cells right here, neurons. After 24 hours, most of them were still alive. He added, there's aluminum that's used in vaccines as an adjuvant. There's neomycin used as an antibiotic. And then here's thimerosal. And as you saw, these did have slightly neurotoxic effects on their own. The thimerosal itself was enormously neurotoxic. After 24 hours, only about 30% of the cells were still alive. Then he wanted to see, well, what if you start combining these things? We know that, for example, mercury and lead are much more toxic together than either one on their own. And he found tremendous synergy so that by the time he mixed all three together, after 24 hours, all of the brain neurons had died. Well, then Dr. Haley asked himself, why this 4-to-1 ratio, boys to girls? What's the difference? One obvious difference is testosterone. Excuse me. So that's what this green arrow is, and when he mixed just thimerosal and testosterone in with the neurons, in 4 hours they had all literally dropped dead. This has been reproduced. It's going to be published hopefully this year. What Dr. Haley also did, and it's not on here, was he put estrogen in the mix and found out that it had a protective effect. And that the neurons actually lived longer. Even though they had thimerosal in the test tube, the estrogen protected, had a neuroprotective effect on the cells, and they stayed along longer. And if you see here, these are 50 nanomolars of mercury. A nanomolar is a millionth of a microgram. So we're talking about tiny, 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 tiny amounts of mercury causing this much damage. I mentioned California. One of the other parents in Safe Minds, Mark Blaxell, figured out the average mercury burden per birth cohort and then compared it with 2 years later the number of cases of autism per 10,000 kids being reported in the California system, which is a smart thing to do. You need at least a 2-year lag time because that's the minimum amount of time it takes for diagnosis to show up. So as you can see, these are kids born in 1989, kids born in 1990, 91, 92. We see a big spike. These were the years that we saw when those big increases in mercury happened. And sure enough, 2 years later, the number of kids presenting with autism was going up in the same percentage as the mercury burden. Now, this again is an ecological study. It's a piece of evidence. It's interesting, but it's certainly not proof. Nothing I'm going to show you today is proof. I specifically titled my book Evidence of Harm, and I'm very thrilled it was just translated into Italian, and I got a copy of the book, and in Italian it's called Prova di danno, which means proof of harm. So the Italians, I guess, are one step ahead of us. They're ready to send in the verdict. You may have come across the term chelation in your studies before. Chelation is the removal of heavy metals using sulfur-based proteins. Again, sulfur has a natural affinity for mercury and lead and other heavy metals. It binds with it and helps us to remove it from the system. This was developed as a treatment for lead poisoning by the United States military back in World War II. Chelation is now starting to be used as an experimental treatment in autism, but it's also been used as a diagnostic tool to try to figure out how much mercury is actually bound up in the system of these kids. You can't just do a hair analysis or a blood test or a urine test and expect to get a reading of the amount of mercury, the body burden in a person, because the mercury is bound up. It's not free-floating unless you have constant, chronic, ongoing exposure. So a kid who received mercury vaccines 3 years ago is not going to present with free-flowing mercury in their blood or urine. You have to give a round of chelation. It's called a provocation challenge to try to get a baseline, to try to see how much mercury you can pull out of that kid. And when we give chelation therapy to autistic children, on average, they dump out almost 6 times more mercury than typical kids. Those typical kids have already excreted the mercury to which they were exposed. The autistic kids did not. In this one, the range, as you can see, was 4.06 to 8.59 versus 1.3 to 1.5 for controls. That was a ratio of about 5.7 times difference. There's a new study about to be published doing the same thing. They found a 5.9 times difference between the autistic kids and the typical kids. This strongly suggests what we call an efflux disorder, a metal metabolism disorder. The typical kids, for whatever reason, their genetic makeup allowed them to get rid of the metals. The autistic kids did not. Another study was done looking at the haircuts of children with autism and typical children. Now, we excrete mercury naturally through our hair, and what they found out was that the autistic kids had very, very low levels of mercury in their hair. The typical kids had huge amounts, and I do hope I find the slide. I apologize. But what was very interesting about it was the typical kids whose mothers had 12 or more amalgams in their mouth, their mercury hair levels were through the roof. I mean, they were beyond the most dangerous levels that you can imagine. But at least those kids were able to excrete that mercury through their hair. There were autistic kids who had mothers with that many amalgams in their mouth, but it didn't matter. Their mercury hair levels were still really, really low. So they were not excreting even that large amount of mercury that they're exposed to. Also, fish consumption in women, it didn't matter. The autistic kids, it didn't matter how much mercury or how much fish their mothers consumed, the mercury levels in their hair were very, very low. Whereas the typical kids, you could almost read, their mother's lifestyle based on the amount of mercury in their hair. The basis of the argument is that we have these class of proteins called thiols, glutathione, metallothionein, cysteine, and they are sulfur-based proteins. And I looked up thiol in the dictionary, and a synonym for thiol is mercaptan, and that comes from the Latin mercurium captans, which means literally mercury capturers. So we now know that kids with autism lack mercury capturers. So when you expose a kid to mercury and they have the inability to excrete it, that mercury is going to go somewhere. A typical kid will excrete it through hair, feces, urine. An atypical kid with an efflux disorder, that mercury is going to get into their brain, it's going to get into their immune system, it's going to get into their gut, and it's probably going to cause all kinds of problems, which we believe may be producing some of these symptoms that we call autism. And Dr. Jill James at the University of Arkansas showed simple blood tests. She tested kids with autism. Guess what? They had little or no thiols in their blood, little or no glutathione. Glutathione is so key. It not only binds with heavy metals and helps us to excrete them from our bodies, it's a very, very powerful antioxidant. And we know that children with autism also have oxidative stress. And Jill James showed that these kids were deficient in glutathione and other proteins. And she devised a treatment to try to get these thiol levels back up to normal, which I'll talk about at the end, because at the end of this presentation, I'm going to be talking about treatments a little bit. So as you can see, actual chart of a child with autism who was given a round of chelation and then measured the amount of micrograms of mercury in their urine. And as you can see, most of the metals are in the reference range, but mercury is so far off the chart, it would go around the wall and probably to the back of the room. In one period, that child dumped out 87 micrograms of mercury, which obviously is a huge amount. Now, here's the marker I talked about, and this is brand new stuff out of France, and it's very interesting. I know Boyd Haley is very excited about this. They took urine samples for French children with autism and found that they had very, very high levels in their urine of something called porphyrins. And porphyrins are used to make heme, which is the oxygen-carrying component in hemoglobin. And it turns out that mercury blocks production of heme, and heavy metals. So therefore, if you have a heavy metal burden in your body, you are going to have a surplus of porphyrins because they're not being used to produce heme. And the French said, we think this is a good marker for mercury toxicity, and what we're finding is that we're finding evidence, is what they wrote, evidence for a link between autism and heavy metals. Now, this is in the Journal of Toxicology and Applied Pharmacology. So that is a peer-reviewed, well-respected journal, and there it is right there in print. And what they looked at, in particular, this one type of porphyrin called coporphyrin, 2.6 times higher in autistic cases than in controls. Now, all of the parents that I know, of course, are running out and testing their kids for porphyrins, and sure enough, they're all way out of the reference range. even some who have chelated their children, they start to see the porphyrin levels come down, but that mercury seems to be so tightly bound into their system that it's going to take a long, long time to get these kids completely cleared out. And here's what it looks like. Autism and autism plus epilepsy are way above the controls, and over here we have Asperger's syndrome. And remember I was saying how maybe they're different than perhaps the environmentally damaged children? This would suggest that you could have Asperger's syndrome and not have mercury toxicity. Now, you could probably have both. There's no reason why you couldn't have Asperger's and also have been exposed to mercury. But in this sample, the autistics were way above controls, whereas the Asperger cases were equal to controls. And then, of course, when they chelated these kids, as you can see, the amount of coporphyrins dropped significantly, as opposed to controls. As a matter of fact, without chelation in the autistic kids, the porphyrin levels went up. So this will hopefully be a brand new test that we can all go out and get a urine test. I think everyone should probably want to see what their mercury burden is. Don't forget, if you're a kid with this efflux disorder, you're going to grow up to be an adult with this efflux disorder. And who knows how many of us might have turned out autistic if we had been exposed to certain chemicals at a certain age. There are people in this room who probably should never ever eat tuna fish or swordfish because they will not excrete it. They don't have mercury capturers, and my guess would be if you tested your porphyrin levels, you would find that they were extremely high. I think this is going to be a very valuable diagnostic tool in the coming years as environmental medicine becomes more and more respected, if you will. Now, I talked about the immune system, and what they found at the University of California, Davis, the Mine Institute, was, again, micromolars, tiny, tiny, tiny exposures for even just a few minutes of time can really wreak havoc on the immune system. They looked at these dendritic cells, and they found that even minuscule amounts of mercury can kill dendritic cells, it can cause cytokine imbalances, and it can induce inflammation. When we look at autistic kids, not their brains, but just their immune systems, we find many of these same things. Increased autoimmunity, extremely elevated levels of cytokines, so there's cytokine expression, and this imbalance in the Th1, Th2 immune response, which has to do with antibodies versus white blood cells and natural killer cells. But their immune systems are completely turned upside down. And we know that mercury can turn immune systems upside down. Then we get to the brain. So you have to realize that mercury will affect different parts of the body in different ways and in different people. We've been looking at the immune system, we've been looking at the nervous system. When we look at the brain, this is where it really starts to get interesting. Dr. Thomas Burbacher, University of Washington Primate Center, also one of the leading toxicologists in this area, has been studying methylmercury in fish and ethylmercury in vaccines. And the conventional wisdom has always been that methylmercury is much more dangerous, that mercury from fish is bad. Mercury from vaccines is not as bad. Ethylmercury clears from the system more rapidly. And Dr. Burbacher found out that that was true. He gave half the primates methylmercury ingested through a tube and half the primates ethylmercury injected, replicating the vaccine schedule. And sure enough, the ethylmercury cleared out of the brain, cleared out of the body more rapidly, and it did not cross into the brain as readily as methylmercury. The press release put out by the government was that mercury in fish is bad, but mercury in vaccines is really not so bad. But that's not what the study showed at all. What the study showed was that once that mercury crossed the blood-brain barrier, because ethyl and methyl are both organic forms of mercury, they're bound with proteins, they can wash into the brain and they can wash out of the brain. They have a half-life of about 30 days. The problem with ethyl mercury is it's highly unstable, and once it gets into the brain, it starts decomposing, and it converts into inorganic mercury. Inorganic mercury does not have that transport mechanism back out. Once it converts, it gets stuck there. It can stay there virtually forever. When we autopsy adults, we find inorganic mercury deposits that are 50 years old that didn't go anywhere. The methyl mercury was much more stable. It did not convert to inorganic at the same rate that the ethyl did. So even though there was more methyl mercury in the brain at autopsy, it had not converted into inorganic mercury. And in fact, the ethyl-exposed apes had 2 to 4 times more inorganic mercury trapped in their brains than the methyl-exposed monkeys. Half of the methyl group had no inorganic mercury whatsoever. So their brains were washing out, but the ethyl-exposed, it was converting and it was staying there. And the reason that's so important is in a previous study done in 1996, Burbacher, the same team, found that inorganic mercury trapped in the brain can spark an autoimmune response by activating certain brain cells called glial cells, astroglial cells and microglial cells. And he found that these were activated. And what's most interesting about these cells is sometimes the activation, the damage, the effect on these cells did not happen until 6 months after exposure of the inorganic mercury trapped in the brain, which might explain why a kid might be exposed prenatally, be born seemingly okay, and then a few months later start to show symptoms. We just don't know. We need to research this much more carefully. But what's interesting is that he found activation of these microglial and astroglial cells caused by inorganic mercury, and he found that ethyl mercury converts to inorganic mercury much more rapidly. Now, to follow that one step further, researchers at Harvard and Johns Hopkins are looking at autistic brains, at autopsy, both children and adults. And at Harvard, Martha Herbert found neuroinflammation, swelling of the nerves in the autistic tissue, and that this suggests chronic disease or an external environmental source, i.e. heavy metals, may be the cause. And then at Johns Hopkins, they found also inflammation, also associated with the brain's immune system. And what they found was that this inflammation was produced by activation of brain cells known as microglia and astroglia. The glial cells, the mercury went right, the autistic brain showed damage to glial cells, activation to glial cells. Burbacher showed that the ethyl mercury converted to inorganic mercury. Inorganic mercury can affect glial cells. Then we have this brand new study. It just came out. Low toxicant levels can damage the brain. Micromolars, again, we're talking about micromolars, whereas we were giving kids, 50 micrograms or more of mercury and lead can lead to developing a damage of the brain cells. They subjected glial progenitor stem cells and at levels that they thought were safe. They assumed that these were going to be safe levels, tiny, tiny, tiny amounts of mercury, and it just shut down these brain cells immediately. Okay, the same brain cells that we're talking about in all the. So what all this tells me is, when we go back and look, some kids, all kids were exposed to mercury. Most kids were able to process that mercury out. They didn't have it in their hair. They didn't have it following chelation. They probably don't have porphyrin levels. But that other group did not. They were not able to excrete that mercury. It had to go somewhere. Some of it damaged their immune system. Some of it might have damaged their gut and their nervous system. Some of it got into their brain, it would appear. And then some of it started doing things like neuroinflammation, cytokine expression, and activation of the glial cells, or some type of effect on these glial cells. If I were a scientist and I had millions of dollars and I had a research agenda, this would be it. This is exactly where I would be looking. But I'm just a journalist, so I just get to talk about it and not do it. Just two other studies that are epidemiological in nature, but certainly would support the mercury hypothesis. This one is very new, out of the San Francisco Bay Area, funded by the CDC. They found out that kids with autism were 50% more likely to live in tracts with the highest levels of chlorinated solvents and heavy metals. And then when they looked at the solvents and the metals, the solvents weren't quite as significant, but the metals were really, really significant. And again, I don't think this is better diagnosis. I don't think doctors in more polluted areas are better at diagnosing autism than doctors in areas with cleaner air. This was published in Environmental Health Perspectives, which is put out by the National Institutes of Health, and is a very interesting study. And again, they say, suggest a potential association between autism and estimated metal concentrations. That's a CDC study. So this is why, and at the end of the talk, we'll get into a little bit more about this move towards environmental causes. Because boy, if we had never put mercury in vaccines in the first place, these people would be all over it. They'd be talking about the coal, and they would be talking about the amalgams, and they would be talking about the fish, but it includes vaccine exposure too. So all of a sudden, this story becomes so much more complicated. And I imagine there are people here who have had it out with the ADA, much the same way that people in my book have had it out with the American Academy of Pediatrics. So there are a lot of parallels. I certainly don't mean to diminish the opposition to the theory that mercury in amalgams may have caused harm as well. But we gave the vaccines to the kids. So that's one reason that thimerosal is such a prime suspect for some of this damage. And then coal also. And we found in Texas, the closer you live to a coal-fired power plant, the higher the chances of your child having autism. So again, it's probably, I believe, going to be total environmental mercury burden coming from a variety of sources, with vaccines being the most common and universal. Now, looking at the evidence that's been presented against it, like I said, it's mostly large population studies. The biggest one done right here in the United States, a CDC study using something called the Vaccine Safety Data Link. This is data at HMOs, mostly in California and Washington. And in the very first run of the numbers, when the CDC did this, they had a brand new researcher there. His name was Thomas Verstraeten. He had come over from Belgium and was not very involved in American vaccine politics. So he just wanted to do good science. He took all the kids, he divided up into kids who got 25 micrograms or more by 1 month, and kids who had no mercury by 1 month. He found that kids in the exposure group were 7.6 times more likely to develop autism than kids in the non-exposure group. That's huge. Anything over 2.0 relative risk is considered causation in a court of law. And as you can see, it wasn't just autism. It was ADHD, ADD, tics, sleep disorders, all of them extremely elevated, and all of them statistically significant. This preliminary version, this study went through 5 different permutations. This is the first of 5. We call it Generation 0 because we found out about it at the end, only because of the Freedom of Information Act. So the CDC was sitting on this data, never ever intending to tell anybody that, at least in their preliminary analyses, they found very significant results from thimerosal exposure in vaccines. And Dr. Verstraeten was so concerned about it, he wrote to his colleagues right before Christmas back in '99 with the subject line, "It just won't go away." They were telling him how to recut the numbers, how to rerun the numbers, how to stratify the kids into smaller and smaller subgroups. By doing so, you break down your statistical power. You lose statistical significance. And it was quite obvious that they were hoping by doing this, they could make that signal go away, that association between thimerosal exposure and negative outcomes. But the signal just wouldn't go away, and they were concerned about it. Now, that tells you a lot about the mindset of what was going on at the CDC. Scientists shouldn't care why the signal's going away or not. Their only concern is it or isn't it? Not, gee, it isn't, and now we need to figure out what to do. This shows a politicization of the process, if you ask me. They're trying to think of other explanations. We can't, the only explanation I can think of is that thimerosal is making these kids sick. What are we going to do? Well, they went through more and more permutations of the study. This was the next generation. This is when they had broken down the kids into different age groups and also by HMO. And as you can see, the rate of autism has come down, but it's still at 2.48. And we have a linear dose response here. And Verstraeten even writes in one of the emails, we do not see any damage until we hit 50 micrograms. So as you can see, these kids who got 50 micrograms, and then 62.5 or over, with every incremental increase in mercury exposure, there was a greater dose-dependent response, a greater linear result towards autism. So this was also of great concern. They took these studies and they kept reworking the numbers. You can see by the next generation of numbers, the rate of autism came down to 1.59, and that line is starting to flatten out. One thing they did is they added younger kids. As kids came into the system, they stuck them into the study. Kids who were 3 months old, 6 months old, a year old. You don't get an autism diagnosis when you're a year old. By adding more and more kids into the population group, they were able to bring down the relative risk. Another thing they did was they initially excluded 25% of the kids because they had other congenital problems, birth defects, whatever. They eliminated them. And one has to wonder, were those kids perhaps more susceptible? Were they more likely to have autism? And as we'll see, they ended up with an ascertainment rate of autism that was way below the California average. Something really bad happened with this study. By the third generation, as you can see, we're now down to 1.52 for autism, and that line is evening out. And then in the final published study in pediatrics they wrote, in no analyses were significant increased risks found for autism or ADD. Well, that really should say, in no analyses meant to see the light of day. Because sure enough, in the first three early analyses, there most certainly were elevated risks, and they were significant, but they weren't meant to be published. And this is just what it looks like graphically. The autism risk went from 7.62 all the way down to nothing. Now, that study, you will often hear people talk about 5 large population studies that prove that thimerosal did not cause autism. 1 in the US, 4 in Europe. This is what the Institute of Medicine relied upon in 2004 when it issued its report saying, mercury does not cause autism, end of story. That chapter is about to open a new page, as we'll see in a minute. But I just wanted to show you 2 of the studies. This is Sweden, and as you can see, Sweden removed mercury from vaccines in 1992, but their autism rate is kind of all over the place with a slightly upward trend. Well, this is only inpatient cases. In Sweden, their registry only tracked patients that were diagnosed in the hospital setting. The vast majority of autism cases are diagnosed outpatient by the family doctor, by the neurological specialist, not in an emergency room setting. So this doesn't really tell us much of anything, and you don't hear people talking about Sweden very much. What you do hear about is Denmark, and the same thing. Here's Denmark, here's the mercury burden, removed the mercury in 1992, and look what happened, the case rates went way up. Well, Denmark also during this period only tracked inpatient autism cases. That's about 13% of the cases in the country. Right around this year, a new clinic opened up in Copenhagen, state-of-the-art clinic. They immediately started diagnosing about 20% of the cases in the country. So the people who run the registry said, well, let's put that data in there too. We should include those kids. And then things were going so well that right around this year, they said, we should just add all cases diagnosed anywhere in the country, no matter what the setting, it should all go into the registry. Because these are only inpatient cases, and then these are then, after the mercury's been removed, they're counting all cases. And what happened was, we went up to 200 cases a year in a country of 6.6 million people. You know, that is not an epidemic. And there are ways to explain 200 cases, even though there's not thimerosal in the vaccines. And that would, of course, might include environmental pollution, fish consumption, coal burning, and immigration. Because don't forget, kids coming from third world countries are vaccinated with the full amount of mercury. And in California, that may actually be keeping the numbers up. 25% of the population in California is foreign-born. Mexico, the vaccination rates are now up to 90%, and they use full-on thimerosal. So Mexican kids coming to this country, we may have people coming to this country not for work, but for services for their children with autism. Just something to think about. And the same thing may be happening in Denmark. But the point of this is this study is often used to prove, they took mercury out and thimerosal went up. Well, the reason it went up is because they started counting all of the cases. In 2000, the very last year, and these are kids born in 1998, look at that. The rate is starting to come down a little bit. Well, mercury didn't all come out of all vaccines in 1992. It was also a gradual removal, just like it was in this country. It took a while for all that stuff to clear off the shelf. So kids born in 1998, kids born in 98 probably, obviously would have received less mercury. We don't know exactly when the last birth cohort was in Denmark, but because they removed the mercury, you would expect maybe the rates to start going down. But these folks knew they had a problem. They submitted this data to JAMA. JAMA rejected it. They submitted this data to the Lancet. The Lancet rejected it. They submitted it to pediatrics. It came back with 5 pages of comments, reviewers' comments. We don't know what they say because they've been redacted. but I know people who have seen access to it. And basically, pediatrics are saying, well, how can you show that this study proves anything, because the rates of autism are starting to come down in 2000 and in 2001. And sure enough, read this email. She says, I need to tell you that the figures in the manuscripts do not include the latest data from 2001, but the incidence and prevalence are still decreasing in 2001. Incidence and prevalence, that means the total number of cases in Denmark was going down. And yet, in the final published study, they didn't include 2001. They had the data. They got rejected from the other publications. Pediatrics finally agreed, or not pediatrics, excuse me. It was pediatrics, because there were 3 different studies. But pediatrics finally agreed to run it, but without the 2001 data in it. So who knows? We don't know. But if this had kept tracking, who knows what 2001 looked like? But if the prevalence rate was going down as well, it's starting to fall. But they chose not to include that in the study. And then, as I said, the IOM finally issued their big report saying we're going to shut this down. Mercury does not cause autism. And they basically based their decision on these population studies that I just showed you. All of that other biological evidence that I showed you had not yet been published. Not Jill James, not Burbacher, not Boyd Haley, none of it. It was presented to the IOM, but they chose to ignore it because they give preference to published studies, fair enough, and they give preference to epidemiology over biology. Well, the biological evidence then had 2 strikes against it. It wasn't yet published. It has since all been published in peer-reviewed journals, and it wasn't epidemiology. So the committee was able to just sort of ignore all of that compelling evidence about what mercury does in the brain, about mercury capturers, about glutathione, et cetera, and concentrated on these large population studies. Even though the federal court system says epidemiology alone is not acceptable to disprove causation, even though the authors of these studies we just looked at admitted that they were flawed, the Danes in their paper wrote that we may have spuriously increased the apparent number of autism cases. There is an understatement of the century. Of course they did. They included the outpatients along with the inpatient and made the numbers go up. Thomas Verstraeten wrote a letter to pediatrics because he was very upset. By the way, he's now safely back in Belgium working for GlaxoSmithKline, and will not answer subpoenas or pesky reporters' phone calls. But he published a letter in Pediatrics basically saying, I don't understand why are you using my study as proof against causation? And he wrote, we found no evidence against an association as a negative study would. On the contrary, additional study is recommended, which is the conclusion to which a neutral study must come. So if you ask the lead author, he'll say, that was a neutral study. We really didn't prove anything. But if you ask the IOM or the CDC or the American Academy of Pediatrics, the well-designed, rigorous controlled study, showed no association. Well, that's not what the lead author is saying. And then finally, the IOM themselves, in their report, buried somewhere on about page 260, they wrote, we cannot rule out, based on the epidemiology, the possibility that vaccines contribute to autism in some small subset. Well, that's what the parents have been saying all along, and that you can't rule it out based on epidemiology, but that's exactly what they did. Now, what's very interesting is Senator Joe Lieberman wanted to set up a panel of experts to go in and look at that CDC data that I showed you, because it's been kept off limits somewhat along the lines of nuclear secrets. And what he managed was to get a special panel appointed by the NIH, headed up by a professor at UC Davis, and they basically critiqued what the government did, what Thomas Verstraeten did. They looked at the data and they looked at the database, and this is what they concluded. They identified several serious problems. There were weaknesses and limitations associated with the database. They were concerned about how autism diagnoses were made. And as you can see, they were very concerned about an under-ascertainment of cases. The state rates at the time were 3 or 4 per 1,000 kids, but in the VSD study, they showed a rate of just 1.1 per 1,000 kids. They missed a lot of kids with autism in the VSD study, and the NIH panel really took them to task for it. They also mentioned the fact that 25% of these kids were excluded and that these same children may represent a susceptible population whose removal might unintentionally reduce the ability to attack. So in other words, this data was bad to begin with, and they handled it very, very badly. This is also very significant. They didn't consider prenatal exposures, such as Rho-D immunoglobulin or the flu vaccine. They didn't consider the cumulative exposure to mercurials through diet or other environmental sources. And they basically said, if you want to use this data to try to, you need to go back to the drawing board and start all over again. So my point is, again, people will point to the US study, they'll point to the Denmark study, they'll point to the IOM pointing to these studies and saying, that's it, this over, and the jury has spoken. Mercury does not cause autism. But they're basing that on highly flawed, highly questionable epidemiology and completely ignoring the fact that these kids are loaded up with mercury. And that at least in some, when we remove the mercury, they at least anecdotally appear to improve symptomatically, some of them. So no matter what the epidemiology says, these kids are sick, they're toxic, they're full of mercury. Does that mean mercury is not the cause of autism? Maybe, but there sure does seem to be an association. So the IRM has kind of reversed itself, and it's going to be a very, very interesting meeting. It's going to happen this April, believe it or not, a workshop to decide on ways to study environmental causes of autism, which is fantastic. Also this June, there's going to be a major trial at the Federal Vaccine Court in Washington, 3-week causation hearing with 3-judge panel that are going to rule whether these cases can be attributed to mercury in vaccines or not. That will be huge as well. But right now, we're sort of at this impasse. We have epidemiology that would suggest no association. We have biology, toxicology, immunology, et cetera, that would suggest there is an association. And where are we going to go with this? Well, just to complicate matters more, we have the question of California. And like I said, California only tracks full-blown DSM-4. So that rise in cases that we saw could not possibly be attributed to better diagnostics, certainly not attributed to PDD and Asperger's, because they were never added on. If your kid has Asperger's, you don't get into the system, you don't get counted. So we saw, as the thimerosal burden went up in California, the case rates went up. Well, the mercury theoretically has come out, and the case rates should therefore start coming down. And for a while, it looked like that's what they were going to do until 2006 came along and just messed everything up. And now it's a real problem, and it's a real problem for this theory. It certainly is a major problem for thimerosal is the only cause of autism. Now, it gets even more complicated because we don't know when all the mercury came out of the vaccines in California and elsewhere. And it's another topic of great contention, and it's something that needs to be settled. The government did not make its announcement until July of 1999. It took drug companies a couple of years to retrofit their factories so that they could then start producing mercury-free vaccine. During that time, they kept producing mercury-containing vaccine. In fact, they produced surplus amounts to provide themselves with a cushion during the transition to mercury-free vaccine. And keep in mind that these vaccines have a shelf life of 3 or 4 years after they leave the factory. That's what the expiration date was. Well, they were still making plenty of mercury-containing vaccine in 2000. They were still making it in 2001. There's reason to believe that they were still making it in 2002. With a 4-year expiration date, there could well be have been mercury, and I have heard people say, they show me the label, DTAP, pediatric dose, expiration date 2006, 25 micrograms of mercury per dose. So there was never just a flip, a switch flipped. And one day we were using all mercury-containing vaccines, and the next day we were using mercury-free. It didn't happen that way. There was never a recall. There was never a ban. This stuff was never pulled from a single doctor's office shelf. And if you're vaccinated at an HMO, where they buy huge quantities of vaccine every year, the turnover rate is going to be even lower, and it's going to sit on the shelf longer. There's evidence to suggest that publicly funded clinics who are receiving mercury-free vaccine during this period. Rather quickly, the CDC started buying as much of the mercury-free as it could. So it may be that if you were vaccinated between 2001 and today, if you were vaccinated at a public health clinic or a military base or a whatever, you may have received mercury-free containing vaccines. But if you were vaccinated at an HMO or a private doctor's office, you may have received mercury-containing vaccines. We just don't know. But California, of course, has the highest HMO enrollment in the country. It's over 50%. And so for kids, it's even higher than that. Most kids in California were vaccinated at HMO. We don't know what kind of vaccine those HMOs were buying in 2001, 2002. We don't know what was stocked on their shelves. And even if you get the lot number, you still would have to go to the manufacturer to find out if that was mercury containing or not. And the drug companies aren't saying. So it's really hard to say, gee, the mercury came out, so the number should have come down by now. We're looking at 3 to 5-year-olds in California. So those were kids born between 2001 and 2003. And it's just impossible to say how much mercury they got. But here's what the numbers look like. And as you can see among the 3 to 5-year-olds, in the quarterly reports, they're kind of all over the place. But every quarter, you get a net gain, which means more kids are coming into the system, into the 3 and 5-year-old category, than are exiting out. Okay, that's bad news. That means the numbers are going up. But as you can see, up until this year, the net change was actually coming down. In other words, there still was a rate of increase, but the rate of increase was falling. There weren't as many in 3 to 5-year-olds. And we just started counting 3 to 5-year-olds in 2003. And so there was falling, and then look what happened in 2006. All of a sudden, we had this explosion of kids in California showing up with autism. So why? What's different about California? Why are the rates going up so high? First of all, we don't know how much mercury was in vaccines between 2001 and 2003. We don't know how much mercury was in Rogam, the immunoglobulin that we were still giving to women during that time. The flu shot was introduced during this period. Pregnant women started getting the flu shot. I don't know if you remember, 2002 was a big flu scare year. A lot of people got the flu shot. SARS was in the news all the time. A lot of people were getting flu shots that had never gotten them before. And then, of course, in 2004, we had the big shortage where everybody was lining up and passing out in the heat in California to get their flu shot. But then we have population and immigration, particularly when I talked about the immigrants. I work with a group in Southern California. They take care of 3,000 families. 10% of them moved to California in the last couple of years to get services for their kids, both from Mexico and from other U.S. states. People go to California to get services. That might make the numbers come up. Environmental burden, environmental mercury, which we're going to talk about in a second, probably is on the increase in California and elsewhere. Then we have early intervention. The CDC is very aggressively going out trying to diagnose kids as early as possible. Could that make the 3 to 5-year-old numbers go up? It's possible. Now, all of this, by the way, I have aluminum in there too because we do use aluminum vaccines. There is some evidence to suggest the amount of aluminum is actually going up because it acts as an adjuvant. It helps spark an immune response to the antigen in the vaccine. Some people hypothesize that the mercury might have been doing the same thing. And by removing the mercury, we're actually making the vaccines less effective. And so in order to get them back up to effectiveness, they increase the aluminum level. Aluminum is also a neurotoxin and it needs to be looked at. This is all conjecture. This is speculation. I cannot stress that enough. The most obvious, easiest explanation is thimerosal had nothing to do with autism. But then the question again becomes, then what is it? If it's not thimerosal, or if it's not mercury, why do these kids have such high levels? And why, again, do some of them seem to improve clinically when we give chelation and other types of treatment? I'm just going to very quickly go through all of these points. This is Dr. Harvey Feinberg. I debated him on Meet the Press in 2005, and he again said the latest vaccines were still in circulation in 2003. So even the head of the IOM says if your kid was vaccinated in 2003, he or she may well have gotten mercury in their shots, and then you have to consider the flu shot, and then you have to consider that seafood warning, by the way, did not come out until 2004. So in 2003, women were still eating plenty of fish. I know women who have autistic children who were never vaccinated. But when you ask them, they said, I ate tuna every single day while I was pregnant and breastfeeding. And I have a mouthful of mercury. Or I had my amalgams removed, right before I got pregnant or during pregnancy. I just got an email from a woman, it was just terrible. She had a very large amalgam removed, and she was breastfeeding, and her 6-month-old son had a stroke and almost died. And, she asked me, was it the mercury? I said, I have no idea. But I do know when you drill an amalgam out of a mouth, and you don't do it properly, those vapors, you just inhale them. One of my best friends was just diagnosed with MS, and that's exactly what happened to her. And when she called me, she had just come back from California, and she told me she was playing tennis one day, and she couldn't look in the sun, and then her legs started getting weak, and she got numbness in her mouth, and she finally had to stop playing. She started seeing double. I said, Patty, you're describing mercury poisoning. I know you think I'm crazy, but that's mercury poisoning. I said, have you had any dental work done? She said, well, yeah, 3 weeks ago I had a really large amalgam removed. And I said, did the dentist take precautions? She said, what do you mean? I said, well, did you breathe through your nose? Was there a water spray? Was there a dental dam? Nothing. And she had inhaled through her mouth the whole time. And of course, when that hot drill bit hits that metal, it just vaporizes. And she now has documented myelin damage in her brain, and they diagnosed her with MS, and she's on once daily injections. And I said my piece to her, but she's going to take the path of treatment that she sees fit. But my point is, we just don't know. My point is, you don't need thimerosal to produce autism, I guess is what my point is. And dental amalgams are certainly one source that needs to be looked at. But up until 2003, we were getting mercury in vaccines. The RhoGAM shot also contained mercury, at least until 2003. The flu shot, I think we've been through this quite a bit, but if I could just stress one more time the effect of that prenatal exposure and how significant it might be. A 1-pound fetus exposed to 25 micrograms is 500 times over the EPA limit. An 8-ounce fetus, the first trimester, which they now recommend for pregnant women, is 1,000 times over the EPA limit. So let's just assume that that's just based on body weight, but we're also talking about stage of development. We know that prenatal exposures are far more dangerous than infant exposures of anything. It doesn't even matter what substance you're talking about. It's more dangerous to expose a fetus to a toxin than it is an infant. So let's just, for wild speculation, it's 4 times. I've heard thousands of times more toxic, but let's just say it's 4 times. That fetus, that 8-ounce fetus, theoretically then is now 1,000 times over the EPA limit. It's 4,000 times over the EPA limit. How can you expect that child to develop normally when he's being slammed with mercury at such an early age of development? And then again, at 6 months, at 7 months, and then the other residual amounts. And we're at 57 micrograms by 7 months, but then we're up to 85 micrograms by 18 months. And the maximum exposure level in the UK was 75 micrograms. This, Mark Blaxell did this chart just to try to show when we're talking about micrograms per kilogram and we're talking about infants versus fetuses, this is the old US schedule, and that was the total mercury burden of micrograms per kilogram. In the old schedule, this is the new schedule with the prenatal flu shot, and in terms of micrograms per kilograms, we're almost up to where we were before. And that might explain why California, one reason, might be going back up. We actually hit almost rock bottom in mercury exposures, and now we're going right back up again, which is insanity if you ask me. So I just quickly want to talk about the environmental stuff because I think it's very, very interesting, and particularly when we're looking at California. Why is California different? What's different? Well, the environment. And California burns no coal, 0, to produce electricity. I believe Arizona is the same. And yet California has one of the highest mercury burdens in the country. A lot of it is coming from China, believe it or not. And in the last 10 years, there's been a huge industrial revolution in China. The amount of coal produced and burned over there has exponentially grown. Every village has its own little power plant, its own little factory. They are zero emissions control. They burn very, very dirty coal, very heavy in sulfur and very heavy in mercury. The amount of coal from Asia has increased exponentially over the years, while the US has actually reduced its coal emissions somewhat, and we all feel good about that. It doesn't matter, because the atmosphere, we're all worried about CO2 and greenhouse gases and everything else. Nobody's talking about global mercury emissions, but they're just as bad and they're just as serious. And what happens is these plumes start to form on the jet stream, and they will take mercury from China, and it crosses the ocean until it hits the west coast of the United States. And depending on where the jet stream is, that mercury will get deposited up and down the west coast or further inland. It basically comes down in the form of rain. So when you have monsoon season here in Arizona, or El Nino in California, or Washington or Oregon, that rain is just washing down all of the mercury that got brought over from China, and this is what it looks like. This is called the Shanghai mercury plume, which I always thought would be a really great cocktail. It sounds like with an umbrella in it. I'll have a Shanghai mercury plume, please. But as you can see, it starts in Asia, and it gets picked up by the winds, and on this particular day, it was deposited in San Diego, California. But it keeps going, and we now know that even in the Midwest, a lot of the mercury, and even on the East Coast, is coming from China. So it actually is circumnavigating the world. Every day, 24 hours a day, more and more and more and more. And this is a very interesting map. This is U.S. mercury contamination from sources here in the country. Now, this is back in 2001. I'm not sure what it looks like today. And it's actually depositions of micrograms per square meter. I think that's so cool. You go out and you measure 1 square meter of land and then measure how many micrograms of mercury fall each year on that meter. As you can see, again, California, the Bay Area, and Los Angeles, pretty high levels. Arizona, crystal clean. The whole mountain state area, when we go back east, and again, Jersey, look at that, very contaminated. Pennsylvania, also one of the highest autism rates in the country, very contaminated. And the West Coast, still pretty bad. But those are just U.S. sources. Now, this is my Al Gore moment. I love doing this because I actually get to be, look at that. This is all sources. So this is foreign sources, mostly Asia. And you can see, look at Arizona, all of a sudden, you're not in clean living anymore. And the Phoenix area in particular, I don't know why, seems to attract a lot of mercury. California is just, look at this, it's most of it's in black or blue, and New Jersey is 100% black. Saturated in mercury. And people say, and I've had this discussion with friends, well, we talk about mercury burden now, and you say it's higher, but what about the Industrial Revolution? What about Dickens? What about the time when everybody burned coal all the time? Well, it just wasn't nearly the amount of coal we're burning now. And again, when you look at it globally, China, 1 billion people, coal is virtually their main source of energy. And look what's happening. These are lakes in Maine. Now, I admit they're in Maine, but if you look at it, they look at sediment deposits. So they can actually go to the bottom of the lake and figure out how much mercury was falling on that lake in 1850 and how much was falling on that lake in 1920, et cetera, et cetera. And look at this. I mean, just take any of these lakes. We'll take this one. This is Lake Goose Lake right here. Or no, let's take, this is the most dramatic one. That's Long Lake. And here, oh, I'm sorry, no, Trout Lake. Don't eat trout from Trout Lake because look at this. It goes up and up and up. Here's 1980. So here's 1990. And here's 2000. I mean, all of these, the peak years are just in the last 5 or 10 years that we really see the mercury levels go through the roof. So pregnant women and small kids are exposed to more mercury now than they ever were before. And kids in particular, they go out in the backyard, they play around in streams, they put dirt in their mouth. They have ways of getting exposed to this, in addition to inhaling the particle contaminants such as a pregnant woman might do, which gets us back to forest fires. And again, someplace like Arizona, where you do have bad wildfires in the summer, and California, all winter long, that mercury might be settling out, and then when it all goes up in flames, again, if you're a pregnant woman living downwind from that fire, which could burn for days, you are inhaling copious amounts of mercury. And they actually did this, and they took forest samples from around the country, and they burned them, and they found 14 to 71 nanograms per gram of fuel. And that's when I figured out how many grams of fuel are there on average per acre. And so it came out to 2.5 ounces of mercury, about 71 million micrograms per acre of forest that goes up in flame. So not to be sneezed at. Then we have crematoria. And California is by far and away the largest user of cremation services in the country. And we know in the UK that 1/6 of all emissions come from crematoria. And this is interesting. As dental care improved in the UK, so did the amount of mercury emissions from crematoria, because more and more people have dental care and have cavities filled, and they're calling it the heavy metal generation, those 40 and above who are now dying with more teeth because of better dental. So it's all your guys' fault. And the average Briton has 2 to 4 grams of mercury in their mouths. 1 gram can pollute a 25-acre lake. Now, I looked at cremation rates in California. Last year, there was something like 110,000 cremations. If you multiply that by 4 grams of mercury, we're talking about pounds and pounds and pounds of mercury going up in the air every year in California alone. And cremation rates are increasing across the country. So just something else. And that's just the tip of the iceberg. I mean, we've talked about forest fires, we've talked about coal, we've talked about cremation, but all these other things, auto junkyards are filled with mercury. All those switches up until about, I think, 1995, all the auto part switches were made with mercury. When those cars decompose, if you live near a junkyard, you're inhaling mercury emissions. Fungicides, when you see those guys out with those tanks on their backs spraying, tree farms, parks, cemeteries, that is ethyl mercury that's used as a fungicide. If you live near that park, you're inhaling ethyl mercury. If you're a pregnant woman, it's going to reach your fetus, or it could. Paper and pulp processors. There are other things in medicine. This is PMA. I think it stands for polymercuric acid or something like that. It's a form of mercury. It's not listed as thimerosal. It's not thimerosal, but it's another form of mercury that's found in biological products. Over-the-counter products. Go right now to the CDC, the FDA website, and look at thimerosal in over-the-counter products. You will get a list that's hundreds and hundreds of products long. Nasal sprays, ear sprays. I mentioned early diagnosis. The age of diagnosis is coming down, which might explain why it's going up in California. I mentioned immigration. I don't have the data for non-California born children entering the system with autism in California, but we do have ethnicity, and you can see the portion of Asian and Hispanic children compared to white children is gone up quite a bit. In fact, they're now at 39% of the total versus 41% of the total of whites. That may be at least somewhat a product of immigration. It would be really interesting to find out. Okay, I'm going to wind down here, but I did want to talk a little bit about treatments, and I am also available for questions all day and night. And I know we didn't get into the legal and political stuff, and it's fascinating, but I kind of wanted to concentrate on science. And when we talk about biomedical interventions for autism, one of the main ones is chelation therapy. And there are a couple of different agents that are being tried for chelation of mercury. The two main ones are DMSA and DMPS. I think it's interesting that the M stands for mercapto, or mercury capturers. Most people have traditionally done DMSA, usually with another chelator called ALA. Others are reporting great success with DMPS, which is typically given transdermally. Now, there is a trial of DMSA that's just completing, first ever chelation trial of autism right here at Arizona State University. Dr. Jim Adams, I understand that it's about to be unblinded. I don't know what the results are. DMSA is considered a less powerful chelator than DMPS, and it was only, I think, a 2 or 3 month trial, whereas typical chelation can last as much as 18 months. So I don't know if we're going to see any difference between the control group and the treatment group. I think we will certainly see more mercury coming out of the control group than the placebo group, is what you would expect. But are we going to see any type of clinical differences in symptoms? The United States government is about to start its own trial of DMSA chelation for autism, the National Institutes of Mental Health. So the next time somebody tells you that this is voodoo and a waste of time, ask them, why is the federal government spending our taxpayer money to see if there's anything to this? If it's voodoo, why are we paying for the trial? And I think it'll be very interesting if there is any significance, any difference between the control group and the treatment group in these studies. That will go a long way to implicating mercury, not necessarily as the cause of autism, but certainly as an aggravating factor, and certainly as something that if kids have high mercury levels, we should do what we can to try to get the mercury out. You may have heard of a horrible case where a child died about a year ago undergoing chelation therapy. That doctor gave the child the completely wrong substance. He gave that child EDTA, but there's 2 types of EDTA. One is typically used to chelate lead, and the other one is used to remove calcium. It's for heart patients. And he gave this poor little boy the calcium chelator, and it killed him. And that's malpractice. It wasn't the chelator's fault, it was the doctor's fault. So when people say, well, a child died from chelation, he didn't die from DMSA or DMPS. I mentioned this earlier on, the work of Jill James, to try to get these levels of glutathione and the body's natural defenses back up to normal. And she's using a cocktail of betaine, folinic acid, and methyl B12. And when I first heard this, I thought, oh boy, that sounds like voodoo. We're going to start treating autism with vitamins. But there is an actual scientific rationale behind it, and it has to do with a very complicated process known as methylation. And methylation occurs billions of times a day. It's the transfer of one methyl group from one molecule to another. And methylation is absolutely critical for a number of healthy processes in our body, including things like nerve development, development of the myelin sheath, development of antioxidants, development of thiol productions, including glutathione, and in fighting oxidative stress. It's also very, very important for the methylation of DNA and RNA for neurotransmitters, which help with memory and attention. And this is what it looks like. And I know you all have medical degrees, so this will be very easy for you. This is what is called the methionine cycle, and this is what is called the transsulfuration pathway. And what happens in a normal, typical person is half of our homocysteine is taken up and it's brought up through the cycle, and this is where methylation occurs. This is where a methyl group is donated to the homocysteine using things like methyl B12. This is methionine synthase, it's an enzyme, and betaine. And what happens is this homocysteine is methylated and it becomes methionine with an extra methyl donor group. The methionine is then sent down this way. Some of those methyl groups are then donated off down here for the methylation of DNA, RNA, membrane phospholipids, and neurotransmitters. So in other words, production of fatty acids that protect our nerves, and the actual neurotransmitters, our synapses, if you will. We need those methyl groups in order to make all of that happen. And these processes are extremely important for memory and attention functions. Then we go back down to homocysteine. I said half of it was taken up and methylated into methionine. The other half is sent down this pathway, converted into cystathionine, cysteine, and eventually ends up as glutathione, the body's natural defenses against mercury and a powerful antioxidant against oxidative stress. Well, what Dr. Richard Deeth at Northeastern University showed was that the introduction of thimerosal in certain children with a certain genetic predisposition can basically block production of MS. The methionine synthase does not get produced in quantities that would allow this extra methyl group to be donated. And what happens is When you introduce thimerosal at this point, according to Dr. Deeth, you basically cut off methylation right there. And once you cut off methylation, you can no longer bring this homocysteine up to methionine. You start producing less and less and less methionine. With less methionine, you have fewer donor groups available for the methylation of DNA, RNA, neurotransmitters, myelin sheath. So again, you block methylation, you block methionine, you reduce the capacity to methylate these important processes. You also greatly reduce the amount of homocysteine available. With less homocysteine, you are not going to produce glutathione. And what you're going to do is, according to this theory, set up a very vicious cycle. Because let's say your first day of life, you have this genetic predisposition, you get hit with 12.5 micrograms in your hepatitis B vaccine. That thimerosal blocks MS production. You shut down methylation from the get-go, and now this kid is not going to have the methyl groups for all these important processes. This kid is not going to be able to produce glutathione, which will lead to oxidative stress. And the next time, 2 months from now, when that kid comes back for those 3 shots, 62.5 micrograms, he's depleted. He or she is depleted. And now it's just going to get worse because there's more mercury and more mercury accumulating. Methylation is shutting down. They're not producing glutathione. They no longer have whatever defenses they originally had, and you're going to get oxidative stress, and you might get memory and attention deficit. So very interesting theory, and what Jill James showed was by introducing this cocktail of methyl B12, betaine, and folinic acid, she was somehow able to jumpstart this process. And she got the methylation going again in these kids, and sure enough, and this is published, peer review, the methionine went right back up. The homocysteine went right back up. The glutathione went right back up to normal levels. And a lot of parents say that when they use this cocktail, sometimes in combination with chelation and or dietary change and or behavioral therapy, ABA, which can work, they see tremendous results. I know kids who have only done chelation. I know kids who have only done chelation and methyl B12. They've had their diagnoses removed. When they went back to the neurologist said, I gave your kid an autism diagnosis. He must have been having a bad day, a year ago, because there's no way this child has autism. And they get their services taken away from them. They get into mainstream classes, and I know some of these kids are now applying for college. They're doing really, really well. And that was apparently due to these types of medical interventions to not only remove the excess mercury, but to try to repair some of the metabolic damage that it may have done. Now, this all needs to be proven. We need so much scientific research, it's not even funny. I'm hoping that money from the Combating Autism Act will be sent to the NIH so that we can finish these kinds of studies and sort of prove this once and for all. But it certainly is a plausible and interesting theory. This one I normally end on, and it's a real downer, but it's important. We still export mercury-containing vaccines all over the world. We are starting to see autism rates go way up in Mexico, in Brazil, in China, in the Middle East, in African countries. And of course, these are countries that a few years ago barely vaccinated at all and are now using full amounts of thimerosal in the vaccines that we export to them. Then on a slightly more positive side, even though the IOM back in 2004 said they weren't going to look at this anymore, these parents have been working so hard behind the scenes in Washington, going to the HHS, talking to government officials, that they got the IOM to agree to a new workshop this April to look at environmental challenges and opportunities for research in autism. And what's very interesting is this was done at the request of HHS and the NIH and the parents. And when the IOM put up their notice, they said that it was be a workshop on the potential relationships between autism and an array of environmental exposures except for vaccinations. It's exactly what it said, and this is last updated on February 2nd. Well, those 3 words, except for vaccinations, have come out. So that explicitly means that they will be studying mercury and vaccines and thimerosal once again over at the IOM, and we have the parents that are in my book to thank for it. And sitting on the committee writing the final report will be Mark Blaxell from Safe Minds and Laura Bono from the National Autism Association, who are great proponents of this theory. So stay tuned. There's going to be a lot more coming up this year. Thank you.